So now the next student is Simay Tuana ALPTEKİN, and she will tell us her story about celiac disease and synthetic interleukin 10. So the stage is yours, Simay.
Thank you, Hocam. I'm trying to share my screen.
So you don't seem nervous. I think you have rehearsed a lot.
I think a bit nervous. I'm a bit nervous.
No need to be. You'll be fine.
Can you see the screen now?
We can.
Okay.
We can see the screen, but it's not full screen.
Now? Is it now?
No, it's still not full screen.
It's full screen for me, but I'm again trying. Is it okay now?
No. Could you reshare your screen, please? And if you do it full screen, that'll be much better. Don't share just your PowerPoint, but share your total screen. All right?
I'm trying. Can you see now?
Yes.
Okay, thank you. Can I start?
Yes, please.
Okay. First of all, hello to my dear professors and friends. I'm Simay Tuana ALPTEKİN, and today I want to explain my project for you. If we come to my presentation content, first I want to start with what is celiac disease, and is there any treatment for celiac disease, and what is the prevalence of celiac disease. Then one by one I will cover each topic. What is celiac disease? Celiac disease is an autoimmune disease which results from a permanent intolerance to dietary gluten, and it is due to massive T-cell mediated immune response to gliadin, which is the main component of gluten. And this is here we can see the prevalence of celiac disease for Europe and Asia populations. And the other question is there any treatment for celiac disease? Actually, until this time, several researches have been done and many treatment methods have been offered. However, for celiac disease, the exact and permanent treatment isn't found. Today, the only appropriate and available treatment for celiac disease is to stop consuming gliadin in our diet. And in patients, it is recommended that strict gluten-free diet can completely restore the intestinal architecture, which is damaged due to celiac disease. But however, we see that even after many years of strict gluten-free diet, celiac disease patients never acquire tolerance to gliadin, and whenever they consume something consisting gliadin, their body reaction is repeated, and these findings suggest that pathogenic T cells remain in the intestine throughout the life in celiac disease patients. And we have research questions, hypotheses, and address problems. The hypothesis: when we apply synthetic interleukin 10 by intravenous to the celiac disease patients with particular frequency, with appropriate dosage, and with uninterrupted treatment process, celiac disease will be alleviated or over. Before explaining the objectives and goals of the projects, I want to explain interleukin 10 basically. It's a type of cytokine actually, and it has an immunosuppressive role, so that we can use interleukin 10 as a treatment in especially T-cell induced autoimmune diseases. And to continue with objectives and goals, the main objective and goal of this project is designing synthetic interleukin-10 with computer softwares, with PyRosetta actually, to prevent natural interleukin-10 serious toxic effects to the body. And we will cover the PyRosetta in later slides. And investigating that whether this synthetic interleukin-10 can be used as an immunosuppressive treatment in celiac disease. Of course, besides that, we want to investigate the relationship between synthetic interleukin-10, regulatory T cells, and also interferons. And also we see that we see the increasing and accumulation of FOXP3 regulatory T cells in the intestinal mucosa of celiac disease patients. And we want to investigate in this project how does it accumulate. And to look at the literature review, from previous literature reviews, we see that natural interleukin-2 treatment received approval for advanced stage of kidney cancer, and it had also side effects to the patients like chill, tremble-like symptoms of flu. And also, again, previous literature reviews, we see that synthetic interleukin-2 was designed in Protein Design Institute, and it's called Neo-215, and it had no side effect to body, and it is totally harmless, and it is used as a treatment method. Also, with the scope of the project, we want to investigate the relationship between interferons and regulatory T cells, to demonstrate whether there is a potentially protective role of interferon gamma producing T helper regulatory cells. And also these are from my literature review. I think to come to originality of the project, we see, I think this project has lots of unique points because the exact mechanism of suppression through FOXP3 regulatory T cells remains uncertain. So this research will be unique for us, and also finding a new solution with interleukin immunosuppressive treatment rather than withdrawal or gliding from the diet or applying strict gluten-free diet. A solution with interleukin immunosuppressive treatment is much more easier for patients, and this will increase patients' welfare. And also, of course, at the end of the project, these proposed ideas both lighten both celiac disease and other autoimmune diseases paths, and provide benefit for patients by alleviating or preventing the disease. And here is a comprehensive figure, and I want to explain it. This is the lumen site, and this is the lamina propria site. We see the microvilli here, and we see small intestine cells here, and we see tight junction between them. And thanks to tight junction, we see paracellular transport here, and with gluten peptide digestion, we can see that gliding particles can pass here. We see gliding specific CD4 T cells here, which we want to measure these in peripheral blood, which we will cover in later slides. And also, as I said previously, T helper 1 type pathway is related to interferon gamma producing. And also we see matrix degradation and mucosal remodeling here because of intestinal damage in celiac disease patients. Also, we see regulatory T cells here, and we can say in cytolysis, we see granzyme and perforin functions in here. To come to research methodology, designing synthetic interleukin-10 is the main aim of the project. This designing will be done by PyRosetta program. The PyRosetta software suite algorithms for computational modeling and analysis of protein structures. Here we can see the how the protein codes are written. We write codes as a command in the software, and we generate outputs from the software. We can see here the accepted energy forms of proteins, the low energy, native energy forms. And also I want to explain these figures. This is PyRosetta Toolkit. We can see the start of region, end of region, and chain ID here. We write the numbers here to generate which type of protein we want, and in here we write sequences here, write codes here, and also below that there is a fold tree, and we can select which edges we want to fold the protein. We have decoy output fat, decoy output name here, and we also have an option of optimize rotamers. We can optimize rotamer with conformational isomerism of proteins. We can see general protocol options here, processor, decoys, and rounds. And the next table, in the first box, we can see the options. Methylation, phosphorylation, acetylation, and sulfation. For example, if we select phosphorylation, in the next box, we have another options from where we want to phosphorylate the protein. Serine phosphorylated, threonine phosphorylated, or tyrosine phosphorylated. We can select one of them. So basically, we select the variant, and we select the code read, and we can see energy function optimization in here. There is electrostatic option here, and we can select one of them and select the statistical or Coulombic electrical charge in proteins. And this is the figure that explains the main logic of actually PyRosettas. First, we have biomolecular modeling problem in here, and then we convert this biomolecular modeling problem to an algorithm. For example, rigid minimization is written here. It helps to minimize the rotational and translational degrees in proteins, and we also have conformer selection here via protein docking, and we convert this algorithm to simulate these in a computer. This is the main logic of PyRosetta. And if we are going to continue, then we have oral wet challenge step. 20 celiac disease patients which have different severity of celiac disease. We collect them 20 celiac disease patients and make them undergo oral wet challenge to stimulate recirculation of gluten specific T cells. And here we can see the gluten specific T cells. And we collect peripheral blood before and after the challenge to measure the severeness of disease for each patient and find out and decide the appropriate dosage of interleukin-10 for each patient.
Simay, sorry for interrupting, but could you please turn your camera on?
I'm sorry. Ekran paylaşmayı tamamladığınızda videonuzu paylaşabilirsiniz diyor ve kameraya basamıyorum ben.
All right then.
Yani basmıyor. Neden öyle oldu bilmiyorum. Tekrar paylaşıp kapatmayı deneyeyim mi?
No, all right then. I was curious why it's closed.
I don't understand those. Can you see now?
Yeah. Presentation, yes, you not. But just continue.
Okay. And we look to other figure. We see the gluten specific T cell response here. This figure is a bit different from the other figure because we see the human proteases and partial digestion of gluten here. And we also, as we mentioned, we also see the interferon gamma producing here. And the other step is applying the synthetic interleukin-10 to patients intravenous or subcutaneous. And after observation, after applying synthetic interleukin-10, we have to observe the patients. And according to these observation results, severeness of the disease and body reactions to interleukin, and the frequency, the applying type, and the next dosage of synthetic interleukin-10 will be decided to continue the treatment process. And the last part is the immunohistochemical double staining for FOXP3 regulatory T cells. When the celiac disease progresses and inflammatory responses are activated, we see that regulatory T cells are accumulated in the affected area. In here, we can see from a research in small intestine and colon, we see the accumulation, and we also see the regulatory T cell accumulation in the other figure. And these are the immunohistochemical double staining for FOXP3 regulatory T cells and CD4 and CD8 double staining images. We do this to double check the decide the severeness of disease and is the patient available to go on the treatment. In work package, in most reasonable conditions, it's planned that the project will proceed at least 1 year, and for every work package, we have success criteria and intermediate outputs. Here is my work package. And before coming to diverse impact, of course, this project needs infrastructure and equipment. For now, we have a computer which has licensed PyRosetta software. We have to have a computer which has licensed Python software, and of course stainings and microscopes for double staining step, and there is a clinical part of this project. I don't want to put these to my presentation because we didn't write them to grant application forms, but I want to mention. In diverse impact, if the project will be successful and proper, designing synthetic interleukin-10 will be an article, we hope that. And also, if it is successful, the software codes of synthetic interleukin-10's domestic and abroad patents will be obtained, and simultaneously phase studies of synthetic interleukin-10 treatments will be done. And I know these steps are much forward steps, as you guess, but I just want to show the general overview of the project. They are not the near steps for us. And there is a social output here. And of course, when the project is successfully completed, we see that treated patients' life qualities will increase. After they eat gluten, their body won't react against the gluten. And for example, when they go to a restaurant or a friend's home, when they consume something, they don't research what's in it, what's the ingredients in it, and their welfare of their life will definitely increase. And also with this project, as a country, we will be in front of the other countries, and it makes a great contribution to our country in healthcare field. And these are my references in the presentation. And thank you for listening to me. I hope I can make a good impact about the project for you.
Thank you, Simay.
Thank you very much.
Thank you.
It was an impressive talk and a very, very fulfilling project. Could you turn on your...
Of course, I'm trying.
Right.
Is it okay?
Yes, now we do see you.
Thank you so much for your comments.
Simay, who are you? Apparently, you're not a first-year student. Are you a PhD student or something like that?
No, thank you so much. I'm so glad to hear that, really.
Well, of course, you are very nervous. It is transferred to us, but even then, it was a very, very fulfilling, and I can see you're very motivated. And I was just thinking that this girl, how can she know too much immunology, like, and coding and like design I'm very impressed.
Thank you so much. I'm really...
Thank you very much. Yes, there can be some comments for improving this, but I don't want to ruin this moment.
Thank you.
It's really nice. Well, by the way, I have some friends who are generating computer models for these biosynthetic molecules. If you want, I can connect you to those.
If it's possible, I really want it.
Right. So once the term is over, come and find me, all right?
Okay.
Right. You could write me anytime.
Thank you so much.
You're welcome. Taner Hoca will comment on something.
Thank you, Ceyhan Hoca. Simay, it is a great presentation. I am thanking you. Actually, I want to ask that maybe certain part I missed. I'm doing experiment at the same time. Have you mentioned for example, the post-translational modifications for generating the actual protein? Well, it might be interleukin, other cytokines, chemokines, it might be. But in order to generate active form of these proteins, in the lectures also, as far as I remember, we mentioned the certain post-translation modifications.
Yes.
Can you comment on which modifications, for example, might be very helpful to generate functional protein? Actually, drug companies have certain secrets about these post-translation modification, because in their lifetime, their experiences have shown that single or very simple these modifications, simple kind of these modifications might really have huge effect. Can you guess which kind of these modifications might have huge impact? Can you guess, or can you explain to us, or
actually,
do you remember?
I mentioned in my grant application form, but I didn't put here in the software. There is also a post-translational modification option. I didn't put here because I can't find the screenshot of the figures, but there is an option like that, and of course it will be done. And in every protein, if I don't remember wrong, we see poly-A binding, and we remove some part of the protein to activate and
For protein also at the RNA level, am I right?
Yes.
In RNA level, if there's a poly-A tail, certain part is discarded, even in the proteins also, in order to generate active form of the protein. This is done. But
yes.
Just I'm only, do remember that it is actually drug company's secret. I just want you to emphasize this.
I can't remember now. Actually, I don't really understand what are you asking, I think.
I can say that I'm just simple asking that glycosylation. Have you ever checked whether you can check the glycosylation of those proteins in order to activate certain modifications also have a huge role. Okay, these modifications is very critical if you want to generate protein.
I remember
protein
it
products.
from collagen synthesis. We have glycosylation steps in hydroxylase residues.
I just wanted to remind that.
Okay,
thank you very much.
Thank you so much for your contributions. Thank you.
Thank you. I am thanking.
Your microphone is muted, Ceyhan Hocam.
Yeah.
But I can understand that you're giving the stage to me.
Yes.
I would like to thank Simay. Thanks a lot, Simay. It was a very good presentation.
Thank you so much.
Thank you, Simay.
It was a distant learning period, and as you also know, all of you, we did not have so much chance to know students one by one and in person. But I could say that Simay was one of those students that I wanted to see as a student, like a questioning and trying to understand the things in the depth and with the ready mind for the high quality discussions, I would say. And now I can see the end product of this kind of a mind in terms of making your project, and I'm very happy to see that. And I would like to send message to all other students is that that's the end of the things that I was trying to push you, like ask questions, criticize the things, don't be afraid of asking questions, and don't be afraid of being on the other side of the instructors, with your instructors. And I know that I write like paragraphs of answers to Simay's questions and then objections. But I'm very happy about that, and that's the kind of the students that I would like to see. I don't want to see a student like nodding to everything, but students like Simay that can object to things. It might be very tiring sometimes, but it's fine. I'm very happy to see that. Thank you very much.
I'm really glad and proud of hearing that, and thank you so much for your contributions.
Congratulations.
Thank you.
Yeah, you think you are proud to hear these, but I
Evet.
admit that we are proud to have students like you.
Thank you so much.
No, I thank you. I'm sure that most of the ideas and the background studies and the planning was done by you, and it is apparent. So I congratulate you. It was a very nice project. Yes, it has to have some optimizations, but well, as I said, you are just a first year student, and just imagining something like this and working on this is priceless. So thanks a lot.
Thank you so much.